In this series of videos, we're going to be taking a detailed look at what enzymes are and what their role is in the human body. Speaking of which, if we were to simplify their role, it would be that they function as the body's biological catalysts, meaning that their job is to speed up the chemical reactions of the body, making sure that they happen in a timely fashion without the need to increase the body's temperature. Simultaneously, if we think about the role of an enzyme in a chemical reaction, if we think of R as the reactants and P as the products, the enzyme would not go on either side, because even though it is involved in catalyzing the reaction, it is not consumed by the reaction, which technically means that enzymes are reusable, giving them tremendous utility in maintaining the body's chemical reactions. Now, this particular enzyme we can see clearly has beta sheets and alpha helical structures, so this enzyme, along with most enzymes in the body, is actually composed of protein, but as we learned in the nucleic acids section, there are examples of enzymes that are made of RNA, and these are what we call the ribozymes, and the ribosome is arguably the most famous example of an RNA-based catalyst. Now, this particular enzyme is a digestive enzyme, and we can kind of see here that it kind of looks like a mouth that is ready to eat something, or if you're old enough to remember who Pac-Man is, it kind of looks like the original 2D Pac-Man, and that actually helps out because this location in here where the quote-unquote mouth would be is actually the most important part of the enzyme, which is what we call the active site of the enzyme. And we call it the active site because this is where the catalysis that the enzyme engages in actually takes place. So this is where specific reactants will enter the enzyme, and then the protein structure of the enzyme will alter in such a way that the reactants are changed in some way and are catalyzed to form the products. Now, when we refer specifically to the reactants that correspond to this enzyme, they are what we call the substrates. So substrate is just another word for reactants, except they refer specifically to the reactants that fit within an enzyme's active site and allow the enzyme to catalyze this substrate in a reaction. And the general idea about each specific enzyme is that the enzyme's active site is designed to be what we call substrate specific. Now, what this generally means, and there are exceptions to this, is that each enzyme's active site generally only recognizes one substrate based on the shape of that substrate, whether it's able to fit into the active site, and perhaps the charge of that substrate, whether the active site possesses a complementary charge that is able to attract the substrate into the active site. And I just realized that I wrote species rather than specific, but hopefully you did not notice that. Now, taking a look at the structure of enzymes in more detail, even though this particular enzyme is entirely made out of protein, many enzymes contain non-protein components, which are generally what we know as cofactors, and there are a variety of forms that cofactors take, many of which are used in the most common and important enzymes and proteins in the body. The first of which, such as this structure that we find here, is what we call a prosthetic group. So, yes, this is the same word prosthetic, referring to artificial limbs, and we use that word to describe substances like this because just like how an artificial limb is not made of the same material as the human body is, this prosthetic group is not made of the same material that composes the overwhelming majority of the enzyme or protein. So prosthetic groups are non-protein components that are part of the structure of the enzyme, and probably the most famous example of a prosthetic group is this structure here, heme, And heme's job is to connect the 4 tertiary structures, in other words, the 4 separate polypeptides that make up hemoglobin together in order to form the quaternary structure of functional hemoglobin. So remember that each color-coded protein is actually a distinct polypeptide, and they are only held together because of these 4 heme prosthetic groups here. In other words, without the prosthetic group, the enzyme or protein, because hemoglobin is not technically an enzyme in this case, the enzyme or protein does not actually form properly, meaning that hemoglobin without heme cannot actually form its quaternary structure. Now, a little bit of technical jargon here. If we were to just look at the protein component of enzymes or quaternary structure proteins, what you would be looking at is what we call the apoenzyme. And if we were to look at the whole structure, including any non-protein based prosthetic groups, you would be looking at what we call the holoenzyme. And this is important depending on what the prosthetic group is actually made out of. Now, the second category of cofactors function very differently. These are what we call coenzymes. So likewise, coenzymes are also non-protein components, but unlike with prosthetic groups, non-protein components are not actually part of the structure. So they are not structurally, let's see if I can spell that correctly, structurally part of the enzyme. So if they aren't structurally part of the enzyme, what exactly do they do? Well, here we can see in a chemical that is involved in the metabolism of alcohol, it requires a cofactor called NAD, which is nicotinamide adenine dinucleotide, which will play a very strong role when we learn about cellular metabolism. So we can see that the enzyme here does not actually fit the substrate ethanol in directly. It actually requires NAD as a cofactor in order to sit in the active site in order to actually help the enzyme catalyze the reaction. So this means that instead of being part of the structure of the enzyme, a coenzyme is actually part of the enzyme's mechanism without actually being part of the physical structure. But nevertheless, coenzymes are completely essential for the process of enzyme catalysis, because even though the enzyme doesn't need it as part of its structure, the enzyme does need a coenzyme as part of the function. And without this NAD coenzyme, this alcohol dehydrogenase enzyme wouldn't be able to carry out its function in order to metabolize alcohol and detoxify it for the body. Now, whether you have heard of prosthetic groups or coenzymes before, you no doubt have eaten many examples of coenzymes and prosthetic groups. So if we take a look at this, probably the most famous coenzyme that exists, we can clearly see that this molecule is organic. So an organic cofactor, organic cofactors such as this one are what we know commonly as vitamins, and if you recognize these as being citrus fruit, you will recognize this molecule as vitamin C, which is an essential coenzyme in the production of the protein collagen that you use to make your tendons, ligaments, and other important bodily structures. Now, dark green vegetables you'll recognize as being an important source of iron. That's what gives it the darker color, and iron is obviously inorganic because it's a metal. So inorganic cofactors, and especially metal-based cofactors, dietary-wise, are what we call your minerals. And iron is probably the most well-known example because we can see that you need iron in the formation of heme that you need to form hemoglobin properly, but there are many other examples. Magnesium is another example of a mineral, and I believe we also need zinc in a number of processes as well. Now that we finished learning about the structure and different components of enzymes, in the next video, we're actually going to look at the general idea behind how enzymes' mechanisms of catalysis work.